And I'm very excited to be joined by Professor Amritpal Mudar. She is a professor of neurosciences at the University of Southampton and has done some exceptional research, which she'll share with us today, and how her journey has been and what she thought she'll do and where she is today. She's also the Dean International. My apologies, my screen is, yes. She's the Associate Dean International and Professor in Neuroscience at the Faculty of Environmental and Life Sciences. And in our last chat, she shared with me how she looks 6 disciplines, all very exciting, and I'm sure she will bring in elements of that in her conversation today. It's very exciting to have you with us, Professor Amritpal, and I'm looking forward to your research on flies and where that has taken you in medical research, right?
Hey, great.
Thank you, and over to you.
Thank you very much, Amrita, for the introduction and for the invitation to talk. And as you've introduced me, I am the Associate Dean International for a faculty that really looks at all aspects of global health, from the health of the seas, the oceans, the earth, the people, psychology, health sciences, biomedical sciences, biology, zoology. But today I'm going to talk to you really about biomedical sciences and my own personal journey. I believe that there will be the opportunity for you to have a similar session like this for the other subjects. And so today I'm just going to focus on biomedical sciences and on neurosciences. And it's very important to really, I mean, this topic, when I was asked to talk about making informed choices, the road less traveled, this was really close to my heart because it's something that I have personally traveled. I've been on this journey myself. As you will see, my aspirations when I was your age led me to make a decision about the university that I chose, the course that I chose, that actually then made me realize that I could get to the same destination, but through a road less traveled, which is what I'm doing. So this is really what I'm going to tell you about. So when I was 18-year-olds don't always know what they want to do, but they know in the bigger picture what they want to aspire to be trying to achieve. And my aspirations as an 18-year-old were too. I wanted to learn about human disease, and I wanted to help humanity in some way. And I thought that that might be through treating disease. And so I was told that you can do that if you become a medical doctor, if you become a physician. I mean, of course, there are allied professions like there's pharmacy, optometry, dentistry. But if you really want to learn about all different aspects of disease and not just the eyes or the mouth, then medicine is what you should follow. And so I started a medical degree. I went to a medical school in London and started in 1991. If I'd stayed there, I would have graduated as a physician in 1997. But within 2 months of being there, I realized that I was very unhappy. I was learning about the human disease, but actually, and where I was going to be able to use that knowledge to treat disease, I was actually not going to be able to learn about the disease in the way that I wanted. As a physician, I would have learned about all of the symptoms that one needs to be able to remember so that you can diagnose the disease. And I was also going to learn about all the medicines one can use to treat the disease, but I wasn't going to be in the privileged position of being able to ask questions about why the disease appeared in the first place. And I couldn't be the one that designs the treatments, that designs the diagnostics to help to treat disease. And after much soul searching, I realized that that ambition I could still achieve, but by doing a different degree. And this is the road less traveled. So I took the very brave, and I don't know if the 52-year-old me now would actually be brave enough to take the decision that the 18-year-old me did. But the 18-year-old me did take the decision to leave medical school, the coveted place that is very hard to get into. And I started a bachelor's of a BSc in biomedical sciences, which then led to a PhD. It says King's College there, but actually, no, my PhD was at the University of Oxford. That's a typo. I always forget to change that. It was a 6-year program, 3 years at King's College in London, followed by 3 years of my PhD at the University of Oxford in Oxford. And I then graduated in 1998 as a scientist, still with the title doctor, but doing something very different. And you now could ask, how does that actually do the same thing? How could I still fulfill my ambitions? If you remember, my ambitions were to learn more about disease and to help humanity potentially by treating disease. I'm still, if I was a physician, I would still do that. Now, take the disease that I work with, Alzheimer's disease. This here is a picture of Alois Alzheimer. She's the first recorded patient of Professor Alois Alzheimer. Sorry, this is, forgive me, this lady is Auguste D., the first recorded case of Alzheimer that was described by Professor Alois Alzheimer in 1906. She has the symptoms of dementia. She has cognitive impairment. She doesn't really know what she's doing and why she's doing it. She looks very vacant in that picture. And on the left-hand side, you see there, I'm try and see if my laser pointer works. Yeah. And on the left-hand side there, you would see what the brain of somebody like Alois, somebody like Auguste would look like. So at the bottom, the brain of a healthy 70-year-old would look something like this. This is where the eyes are. This is the back of the brain. But somebody like Auguste D., she would have a brain that looks like this. That's all shriveled up, has lots and lots of cell death. And that's why she's got symptoms of learning and memory, especially cell death in this area where you deal with early memories, what you've done here and now. That's what was first in the people who have Alzheimer's disease. So as a physician, I would be able to diagnose her condition as Alzheimer's based on the clinical symptoms that I will have learnt in my medical training. I would have been able to treat the disease based on historical information that has been delivered to me through my medical training, as well as through my own practice for what I know works to treat people like that. So I would still be able to learn about disease and treat disease and help humanity. But that didn't really satisfy me. However, as a scientist, if we take the same disease, I can do something different. So this is the road less traveled. As a scientist, I get to ask questions about why and how the disease appears in the first place. I get to ask questions about how the cells are dying, why they're dying, and why does it happen to people who are in their 60s and 70s and 80s? Why doesn't it happen to young people, even if they've got a genetic predisposition to the disease? And I can then use that information to create novel drugs to treat the disease. And I can also use this information to diagnose the disease, which is very important because, as with all conditions, if you catch the disease early, you're much more likely to have an effective treatment. It's like that saying, a stitch in time saves 9. If you catch any disease very, very early, you're much more likely to treat it. And with some conditions like dementia, the clinical symptoms appear 20 years after the disease started. So it's very difficult at that time to treat the patient with anything that will save the nerve cells because they're long dead. So you need to really understand how they're dying so that you can pick up symptoms of that cell death very early to diagnose the disease early. And this is really what the scientist does. And this is what I did. And I want to give you more information about how I achieved that over the last 25 years so that you can see how the road less traveled still takes you to the same destination. It's just that you may enjoy that journey more, you may enjoy what you do more, and it may be something that inspires you more. So how does the scientist actually do this? How does the scientist ask questions about how and why the disease appears? So to remind you again, in Alzheimer's disease, you've got memory loss and you've got cell death. Alois Alzheimer looked deeply in the brains of people who have this Alzheimer's disease and was able to describe the appearance of these structures, these dark structures. This is a section through a brain. These are nerve cells. Those of you who are doing A-level biology will recognize this. In fact, GCSE biology students should also recognize these nerve cells. But these dark structures here, they are tangles. Tangles are rope-like structures that just grow and clump inside nerve cells, and they just make those nerve cells sick. They just kill the nerve cells because they make them very, very sick. As a scientist, I can ask many questions about this disease. And the first question that I asked was, what are these tangles made up of? And this wasn't work done in my lab. This was work done by my predecessors in other labs in the 1970s, where people found that these tangles are made up of a protein called tau. Tau is a normal protein. We all have that protein. It's in every nerve cell in our brain. But this tau protein becomes abnormal in the brains of people who have Alzheimer's disease. And then it starts to become sticky and it starts to clump together and it starts to really grow inside those nerve cells and then kill those nerve cells. So as a scientist, you have the liberty to ask any questions that you like about this process. So I'm going to pause for a second and I'm going to ask you, what question would you ask as a scientist? And I don't know, Amrita, how they will answer the question, but I'm asking the question now. How, if you guys were the scientist and I've given you the information I've given you, what kind of questions would you ask? And I presume they can put their answers on a chat. You'll have to read the answer questions for me because I can't see the chat. So the question is, what kinds of questions would you ask armed with the knowledge I've given you? Sonali or Amrita, are one of you there? Are you able to tell me if this engagement is okay? I don't hear from any of you, so I don't really know. Amrita, can you tell me if people are, because I can't see the chat. So are people putting?
Sorry, hi. I can read out
Yeah.
questions. I'm just waiting to see if the students have posted anything right now.
Okay.
Um,
Yeah. So the question is, how would, armed with this question, armed with this knowledge, what kinds of questions would a scientist ask? If you were a scientist, what kinds of questions would you ask?
I don't see anything from students right now.
Okay, I'll carry on then, but they should be primed to be able to answer questions as we go forward. I would ask the question, how does the normal protein that we have in our brains become abnormal? Number one. I would also want to ask, how does it actually make those nerve cells sick? If it's a normal protein, it's clearly doing something. When it gets sick, what happens to the nerve cells? How does it make the nerve cells sick? The second question I would ask is, how does it kill the nerve cells? When the protein is sick, the cells are becoming dysfunctional in some way. How do they then die? And my third question would be, how can we stop that process to actually treat the patient? Or how can we detect the process early so that we can pick up the individuals that have this process happening in their brains at an early stage? So those are the kinds of questions that a scientist can ask. And certainly they're the kinds of questions that I was trained to ask when I did my PhD, because I did my PhD in Alzheimer's disease in the University of Oxford in 1998. I graduated. And since that time, including the time when I was in Oxford, and since that time, I have been asking the same questions in my lab. So that's, sorry, the last thing there. How can you actually use this information to diagnose and treat patients? So how can a scientist ask these questions? The first thing you do is you recreate an aspect of the disease in an animal model. So in this case, we recreated the abnormal tau inside nerve cells of the fruit fly. And oh, just want to pause for a second there to tell you why I thought the fruit fly was a good model organism within which to ask that question. Because when you recreate an aspect of the disease in an animal model, you then test your hypothesis. And my hypothesis was that when this tau protein is abnormal in the nerve cells, it's actually not being able to do its normal job properly. And if it can't do its normal job properly, then that's how the nerve cells are getting sick. That was really my hypothesis. And I chose to test that hypothesis in fruit flies. So fruit flies are they've been used in medical research for a very, very long time. They can be manipulated very easily, and you can ask questions about any aspect of biology in these cells. So again, I pose another question. Why do you think it's a good idea? Why do you think I did this experiment of testing how abnormal tau makes a nerve cell sick? Why did I do that in the brain of a fruit fly? Why didn't I do that in a nerve cell that I grew in a dish? So again, Kasvi, if you can read out to me anything in the chat. So please don't be shy. You obviously don't know these answers because you didn't know the questions before you appeared in the chat, in the forum. But it's your opportunity to really stretch your mind. So think about it. Why would I recreate abnormal tau in the brain of a fruit fly rather than recreate it in a cell in a dish if I wanted to know whether or not it can't do its normal job properly, and that's how the nerve cell gets sick? So we'll wait again to see if the
So we
individuals
have
are
a few responses. I can
good.
read them out to you.
Yeah.
We have one from Lakshay saying that because there are fewer neurons.
Okay, that's a
We
very
have
good idea. If there are fewer neurons, then that's the reason to do it in a fruit fly rather than in a rodent, so you can study them better because there's fewer of them. Yes, that's good. Anything else?
we have one from Hidvi saying fruit flies have simple structures.
Yes, that's also
Um,
true. And in fact, I could visualize the nerve cells very easily in fruit flies because their structures are simpler than if I looked at circuits in the rodent brain. Anything else? Anything about why not do it in a cell in a dish?
So Tisha says so it can live in a real organism.
Excellent. You want to really do an experiment in a whole organism so that you can look at the impact on behavior. If you recreated abnormal tau in a nerve cell in a dish, that nerve cell in a dish isn't going to engage in learning and memory. That's only going to do simple cellular activities. So if you wanted to see how that nerve cell becomes sick and how that affects the behavior of the animal, you really need to do it in a whole animal. So thank you very much, Kasvi, for reading that. So our hypothesis
There's
then
actually
was,
one more
yeah, go ahead.
from Amrita, and Amrita says that so that it's not in an isolated setting, which is something that you've already, so that it's not in an isolated setting, which is something
That's
that you've
right.
sort of already answered.
Yes, absolutely. Because we want to see how those nerve cells are working together to give rise to the learning and memory in people, and how when one circuit gets sick, how does the overall learning and memory get affected? And of course, if you want to then do drug treatment and you want to see whether the drug improves learning and memory, again, you need to do it in a whole organism. So thank you very much. Those were excellent suggestions and exactly what I would have expected from such a high achieving group. Amrita told me that there's a very high achieving group I'll be talking to. So I'm very pleased with your input here. So the first question we have to ask ourselves is, in order to understand how the nerve cell gets sick when the tau becomes abnormal, we need to recreate the abnormal tau in a circuit, and we've chosen to recreate it in the circuit of a nerve cell in the fruit fly brain. But we have to have some understanding of what the tau normally does in the nerve cells to then have an idea what might go wrong when the cell gets sick and the tau becomes abnormal. So this here on the left-hand side is a simple diagram of a neuron. Here is the cell body, which is the housekeeping hub where day-to-day activities take place. Things are synthesized. Many other processes take place here. Whatever is synthesized here is transported down this highway that is called the axon to the synapse, which is the business end. The synapse is what communicates with other regions that circuit wants to engage with. So learning and memory will have many, many circuits working together where circuits will communicate with each other. Information in the form of neurotransmitters will be synthesized here, transported down the axon all the way down to the synapse. But if you look at higher magnification in the axon, you see the appearance of tracks. They really do look like rail tracks. They're long structures made up of polymers called microtubules, but these polymers always break down. They need to be kept stable with proteins like tau that really sellotape it and keep them intact so that cargo can be transported down these tracks. So my next question to you is, when tau becomes abnormal, as it does in Alzheimer's disease, what do you predict will happen to the function of tau? And here the function of tau is to stabilize the tracks, provide transport for vital materials within the nerve cell. So if you can tell me, Kasvi, what students think happens when the tau becomes abnormal.
Absolutely. I will give it a few seconds. We just have one response right now from Hitvi saying axons will start breaking apart.
The axon, so the axon is what contains the tracks. So it's not the axon that will start breaking apart. The tracks inside the axons will start breaking apart. Anything else? What do you think will happen to the transport?
We have one from Lakshay who says the microtubules aren't stable as TAU does not hold it together.
Very good. The microtubules absolutely won't be stable because the tracks, because the tau is not holding it together. So then, as the previous student had said, the tracks will fall apart and the transport will essentially break down. That was our hypothesis. And so we tested this hypothesis. So the scientist would recreate the abnormal tau in the circuit in the brain of an animal and then test to see if all three of these things are happening. And so if you look inside the animal model that we created, all of those three things seem to have happened. First of all, this here are the nerve cells of the fruit fly. And in these nerve cells, this is a control animal that's a healthy animal. And there's a green fluorescent tag that has been inserted into the cargo. So you can visualize the cargo moving up and down. This is in a control animal, an animal that really only has the fluorescent tag in the cargo and it doesn't have any abnormal tau. This animal has normal tau. This animal has abnormal tau. And you see that the cargo now starts to get clumped up along the axon instead of being spread out all the way along the axon. What do you think this is indicative of? What does this indicate? The fact that the cargo in the presence, this animal has the abnormal tau, this animal has the normal tau. The cargo here is spread out, distributed all the way along the axon. The cargo here is clumping. What do you think this is indicative of?
So we have a few responses. We have clumping and synaptic dysfunction.
You think there might be synaptic dysfunction? Yes, but tell me, what does the clumping suggest?
So they're saying this could lead to tangles.
It could lead to tangles, but the clumping here is the cargo that's clumping. But yes, the tau, this is here a marker of the tau. The tau here is looking like it's also clumping. So this could definitely lead to tangles. The students are right. But these clumping of the cargo is suggesting that axonal transport is disrupted. And we wanted to test the behavior of this animal. We did look at the synapse and the synapse was also dysfunctional, but we wanted to look at the behavior of these animals. And we did a very simple assay, which I call the writing assay. And again, this goes back to the question I asked before about why would you do this in an animal? Why would you not do it in a dish? And we said that the animal can engage in behavior. And the behavior here we were looking at was the ability of this animal to write itself. So these are all of these are kind of control animals, and what we were doing with these animals was we were expressing the tau in their circuits. We were then rolling them onto their back. They don't like being on their back. They use their nerves to engage in a reflex action that rights themselves, that helps them turn around, and we call that the writing assay. So control animals, these three bars here, if you look at this particular one, this control had no abnormal tau at all. This animal only had normal tau. this animal had the very nice distribution of vesicles, and this animal, when you roll it onto its back, rights itself in 10 seconds. This animal here had this kind of clumping. This animal had lots of abnormal tau, and this animal takes, as you can see, almost 5 to 6 times longer to right itself, which suggests that when you've got this disruption of transport inside a circuit and material doesn't get to the synapse can't communicate with its target, and that could be why that circuit doesn't behave as it should. You get that level of dysfunction, that level of impairment. The circuit is still alive, but it's sick. And this might be happening in the brains of people who have Alzheimer's disease at the earlier stages. They don't really remember things quickly. They don't learn things quickly because the circuits within which the tau is becoming abnormal, within which the tau is clumping, axonal transport is not occurring. There, you may not have effective communication between the circuits, and their behavior will definitely be impaired. So we think that this is happening because of what? So again, question to the audience. Why do you think this is happening?
We haven't received anything in the chat box yet, but I will give everyone a few seconds to respond.
Yeah, I'll repeat the question. We have the disruption of transport. According to our hypothesis, why do you think the transport is disrupted?
So we have our first response, which is from
Yeah.
Tisha, who says the vital materials are not being transported, hence their functions, which normally would help, is not being done.
That's the reason why we have the behavior, absolutely right. But why do you think the materials are not being transported? What do you think is happening in the nerve cell for the transport not to occur?
So Lakshya says because the neurons can't communicate with each other as the cargo is clumping and the microtubule is broken.
The microtubules are broken, absolutely. We think that the cargo cannot get through because the microtubules are broken. So we looked using electron microscopy to see if the microtubules were indeed broken. And this is a nerve bundle. You imagine it's coming out of the screen, and we've sliced it in cross-section. And these little round circles that are in the axon, they represent intact microtubules that are stable. They are stable, which is why when you slice it through, they actually look like little round cylindrical structures. And these are the ones that you see in normal animals where the tau is healthy. This is what you see in animals where the tau is unhealthy. There actually is virtually no intact microtubule. All of these structures here are not appearing there, which tells you that absolutely, as Lakshya suggested, the microtubules are broken. And indeed, this is very clear evidence that the microtubules which look like this are looking like this. So no wonder the cargo is getting clumped. No wonder the cargo doesn't get to the synapse. No wonder communication is so badly disrupted. So now I want to ask, how do you think we can use this knowledge to devise a therapy? So again, others in the audience tell me, you have all this information about how the cell has got sick. You, the scientist working on a human disease, have learned about why the cells are getting sick in the human disease. How can you use that information now to create a treatment? What kind of treatment do you think you can create?
So we have our first response, which is genetic modification or healthy TAU injection. And then we have promoting microtubule acylation.
So you could promote microtubule stabilization, yes. I think the first response about genetic modification, I think, would be difficult. What would you genetically modify? But the second response is very interesting. that now can you know that the microtubules have broken down because the tau is not doing what it should do. Can you promote the microtubules to be stabilized in another way? So we thought, can we give the animals drugs that actually put those microtubule tracks together? And in fact, there are drugs, chemotherapy drugs actually work by overstabilizing microtubules. So we treated these animals with a drug that could potentially put the microtubules back together. This is an animal that had tau, that actually had the abnormal tau, and there were no microtubules there. And this was an animal, a sibling of this animal, who had been given this drug, this microtubule stabilizing drug, for only 24 hours. And you got a complete rescue of the microtubules so that they are no longer destabilized at all. In fact, they're completely put together. So it's very, very clear evidence that if you know how the nerve cell gets sick, you can actually provide the animal with a treatment that can actually reverse the cause of the sickness. And this was really now what the how my aspirations over the last 20 odd years have unfolded. So my aspirations as an 18-year-old had been to learn about human disease, to help humanity by treating disease. My aspirations as a 50-something-year-old are still the same. I still want to learn about Alzheimer's disease. I still want to treat it. And I've only told you about one experiment and one project where you can see how through this road less traveled, I was able to do this, but through a different road less traveled. So not through the normal route that everybody takes, but through this other route. And over these years, I have modeled aspects of the disease in rodents, in flies. I've been able to identify, this was the first drug that I told you about, the drug that was given here, this drug that completely rescued the problems in the microtubules. We published that paper in 2013. That drug was given to rodents. It was given to people too, but there was a reason. I'm going to ask you guys what the reason was why it will not have worked. But just to complete the point I was making in this slide, we've then carried on working with these models and identified 2 different drug targets over the course of the years, and now we are starting to work on diagnostics where we can pick up symptoms of the things going wrong very early. But coming back to this slide, given that it worked so effectively, this drug was given to people with mild cognitive impairment as an initial trial, and it worked very nicely in those people. But then the drug company got very greedy and decided to give it to people who had very severe forms of Alzheimer's disease, and the drug didn't work. Can you tell me why that would have been the case? So why do you think the drug wouldn't have worked if it was given to people in very severe forms of disease?
So we started getting a few responses,
Mhm.
and some of them are maybe because the neurons are already dead.
Spot on. Your first response is spot on. In people who have a very severe form of disease, they have already been through this stage. They've got long past this stage. The neurons were sick, and very quickly, over years of being sick, they then eventually died. That cargo never got to the parts of the nerve that needed the cargo. That nutrition that the rest of the nerve needed never got there. And after a while, after a time, the nerve cell just died. And then it doesn't matter that you're giving it the microtubule stabilizing drugs. There are no neurons to save. And that was really why that clinical trial failed. And the drug company lost so much money that they couldn't then put that drug into another clinical trial later. And I'm told by the CEO of that company that they're now looking for new investments, and I think have found a new investor who will give permission to try this drug again. But this time they're going to select people that are in the early stage of disease rather than testing it on people in the later stage of disease. Do you want to tell me any other reason, and before I move on to the next slide?
So we have two more. One
Yeah.
is that maybe it wasn't enough to completely fix the problem, and it's possible it could interfere with the normal function of the TAU.
The tau. Yes. So those are true as well, that it could be that there wasn't enough drug that was given, but the drug companies have done those tests before they get to this stage. They know the dose that is adequate for penetrance into the brain. So they know when the dose is not enough, because they just give you more to see if it's effective. It's more the fact that you really were giving it to people where those circuits were completely dead. So there was really no scope for reviving the transport, because the neurons are long dead, which is why it becomes very important to create diagnostic tests that can pick people up when the nerve cells are just sick, they're not completely dead, because then therapies like this one will work. And so this was really just one example of the kind of thing that someone with a degree in biomedical sciences will do. So if you do a degree in biomedical sciences or a degree in biological sciences, you can do the kind of research that I've described to you, which will allow you to learn more about disease and actually treat patients in a different way. You will be finding the treatments that the doctors then get to be able to treat the patients. But there are many other professions where you can do that. So industry, biomedical research, which is what I've described to you about, clinical research, they're all arms of different aspects of the same type of work. So somebody working in industry would then take the drug and start working on making the drug more able to enter the brain. Biomedical research is the type of work I've described. Clinical research would be where the scientist works with doctors to be able to test whether or not the drug works in clinical trials. And I've given you this example from the point of view of neurosciences, but this could well work from cancer biology, any other ailment, every ailment that the doctor treats. Actually, there is a scientist behind that doctor that has worked out how that particular ailment arose and what treatments can help. And then you can have the people in pharma who create the medicines and people who do the clinical trials. But all treatments have to then also be protected by patent law. So many people with a degree in biological sciences actually go on to become patent law attorneys. So these attorneys don't really need to have a degree in law. In fact, you won't have any patent law attorneys with a degree in law. They will have a degree in a biological sciences subject, and they will then be able to go on and do a degree in law. The company will pay for hands-on training in patent law for them to then be able to apply that knowledge in patent law. And what the patent law does is he or she ensures that the drug you've created hasn't been created by anybody else before, or other people later can't copy that drug and then use it to treat patients. That becomes your intellectual property. But they need to have knowledge of the biology that led to the drug discovery before they can actually apply the patent law to it. Of course, you've got other people that don't work necessarily on medicine and medical related aspects of biology. They look at other things. So you've got forensics, where a lot of your knowledge about the body helps you in forensics. In, for example, working at the time of death, you need to have a very strong degree in biology and biochemistry to be able to see how the degree of decomposition of a body, for example, gives you information about the time of death, and whether how DNA testing allows you to work out whether XYZ was at the crime scene when the murder or whatever you're investigating happened. Of course, you've got biologists who work in agriculture and environment, and a lot of what they do is actually fascinating. So, for example, a lot of work is done on GMO crops that enable the crops to be genetically modified so that they are resistant to pests. There are other, some of the work that we do in our department is, I'd never believed that this would happen, but it's amazing. They create genetically modified crops that are not genetically modified themselves, But what they do is they are able to, they're genetically modified to enable them to be better hosts for bacteria. And these bacteria are friendly bacteria. But when these bacteria grow on those genetically modified plants, they repel pests. So the bacteria almost becomes like your security guard. So the genetically modified plants are able to harbor security guard type healthy bacteria that then ward off other harmful pathogens for the plant. That's an example of how knowledge of biology actually can be used to help mankind in a different way. And I've put there business, teaching, and science communication, which again are vital areas that people with a degree in biological sciences go into. And you'd never understand, why does somebody with a degree in biological sciences go into business? What can they do for the business people? We've had some of our graduates who've studied biology or zoology employed by JP Morgan or other high caliber banks, because they want, especially now going forward, they want to serve their clients with ethical, they want to make sure that a lot of their decisions are not just financially sound, they're also ethically sound, they're environmentally sound, they're sustainable for the environment, they're sustainable for the ecosystem where that business is going to be trying to make money. So for example, one of our zoologists works on building sites with businesses where she has to ensure that the ecosystem around the business is told about the impact of building that building there on the ecosystem nearby, so that the company doesn't actually do something that is good for the company but really bad for the environment. And those kinds of that kind of information isn't going to come from a business degree. It's going to come from someone who's got a degree in biological sciences. So that's one more example. And then science communication. We've all lived through COVID. A lot of what we learned every day on the news was through people who communicated with us. And those people weren't the English graduates, they weren't the law graduates, they were the science graduates who needed to understand how scientific information that needed to be factually correctly conveyed to audiences was conveyed not just in a way that it was factually correct, but in a way that it was clear for them to understand. And that again requires a degree in a biological sciences subject. So a degree in biological sciences really can take you many, many places. And then you might ask me, what actually are degrees in biological sciences? And these are my last two slides. So I've told you about neurosciences. Many universities offer degrees in biological sciences, and you get confused. What does that really mean? What are the different degrees within that? Biological sciences, in a nutshell, is really the study of life. It's the study of life at the level of the molecules and the cells and the tissues that make up the organism and how that organism interacts with its environment. If you studied something like biology, you would study all of these aspects in plants and animals. And in Southampton, our biology degrees are very, very, very flexible. So you can focus on any of these areas more if you are more interested in them. We also offer a degree in zoology where you would study all of these relationships. You'd study life at all of these levels, but primarily in animals. If you were interested really in the nervous system, then you would study the molecules within nerves, how the neurons interact with each other, how the brain interacts with the environment and with the rest of the body, and how that controls the organism. So your study of life would be really in all aspects of the nervous system. If you are interested in biochemistry, for example, then you'd be really focusing more on molecules and cells and how behavior of molecules within cells really helps the organism function, but your primary goal would be in molecules and cells. If you studied pharmacology, for instance, then you'd be looking at how drugs interfere with molecules, how that alters the behavior of the cells, how that influences tissues and the organism overall. And so there are many different topics within biological sciences. We have one more new degree that's coming out next year, this current year, in microbiology, which we've added to that list. And you can always study these at undergraduate level or master's levels. And then finally, the question would be, why would you study biological sciences? For me, It is interesting, it's challenging, it's exciting, but more important of all, it's rewarding. The road less traveled is taking a route that enables you to do all of these things, but in a way that you may not have thought about before. And I have spoken for a bit longer than I needed to, but we had the interaction too. But we now have about 13 minutes, I believe, for questions. And I'm very happy to take questions in every aspect of my talk. I've got my email there too, but I can answer questions about my talk. I can answer questions about other degree programs, why I chose what I did, what I do now. Yeah, so over to you.
Thank you. Thank you, Professor Amritpal. I think it was very, very eye-opening, and it took me back to the physiology of the brain and the biology of psychology, what I had studied back in school, though I wasn't as sharp to give you the answers that the students here could, but thank you for spending time and sharing about your research. I want to encourage all students to ask their questions, and I'll start with the ones that are already in the Q&A box or in the chat. So, and Nadeem, if you could respond to the questions that are related to the applications and admissions, then we could focus more on the ones around research and programs with the professor. Professor Amritpal, the first question is from Tisha. Does the University of Southampton offer forensic science courses?
So first of all, so the short answer is no, we don't. But the longer answer and more relevant to you, Trisha, if you were my daughter or my personal tutee, I would not advise you to do a degree in forensic sciences. I would advise you to do a degree in biochemistry or in biomedical sciences. And then I would advise you to do a master's in forensic science, because forensic science is an application of a subject. You should really become very proficient in the subject first, and then do a master's in that subject. It's like if you wanted to learn a language, you don't just go and get text and then learn how to write the text. You actually learn the alphabet, then you can write any text you like. And that's really very good, a very, very good idea to have a very broad base. Allow your degree to be broad enough for it to be applied to anything you want. And the second reason to do that also is, after 3 years, if you did forensics and you realized, actually, do you know, I like it, but not as much as I thought I would, you are not able to do anything else. Whereas if you did biomedical sciences, you can still do forensics, and you'll be better forensic biologist, but you can also change your mind and do something else.
Thank you. That's a very response that allows students to have a broader vision and a long-term perspective and longevity of their career and ability to sort of take twists and turns as they go along and not get boxed very early. Thank you for sharing that. Melanie actually asks a question on how does biotechnology differ from biomedical sciences?
Again, a very good question. So biotechnology, it could come into the realm of industry, because industry is using science to be able to provide solutions for many different problems. So biotechnology could be in the creation of diagnostics, for example. So these COVID tests that have come from biotechnological innovations, it's not just knowing how you can capture the antigens or bits of pathogens that are floating about in biofluids. How can you capture them? It's also knowing how you can, you're not going to really take during COVID, we didn't have great big machines that went to people's houses. We had little tests that went, these little small discs that we had, that everybody had. That's also biotechnology. That's understanding how you can miniaturize things, how you can make them accessible to the community at large. So biotechnology is many, many different things. Another aspect of biotechnology, for example, totally different to biomedical sciences, is use of restriction enzymes that could be used in many different ways. So restriction enzymes can be used for diagnostics. Restriction enzymes basically just cut DNA at different places. And if you cut DNA of bacteria, for example, with 2 restriction enzymes, the size of the cut you'll get will give you an indication of the type of bacteria that was in your sample. You might want to use that not just to diagnose disease, you might want to use that to look for purity of water, to check for a salmonella infection. You could look for many other things in different scenarios. So biotechnology is very broad. And again, if you wanted to do biotechnology, I would strongly recommend you do a broad degree in biology or biomedical sciences, depending on whether you want to do biotech within the health realm or biotech in any aspect. So detergents, for example, you know, when you have biodetergents, they actually have enzymes in them that digest the food. That's how they remove the stains from the food. That's another example of biotechnology. So you can think about the type of biotech you want to go into. And again, I would do a broad degree where the modules allow you to take some modules that are specializing in biotech. Then I would do a master's degree in biotech.
Okay. We have some more questions. So Hetvi asks how, there are lots of questions, so bear with me. Hetvi asks, how equipped do biotech courses in University of Southampton make the student for practice, say in other parts of the world? Is there any limitation that studying biotech in Southampton may lead to difficulties practicing, say, in India?
So we don't offer a degree in biotechnology, just as we don't offer a degree in forensics, but for the same reason, because biotechnology is an application of knowledge. And so we will have aspects, modules within our degree programs where you can think of applying your knowledge in a biotech sphere. And we also have the ability for you to be able to do modules that are in other faculties. So biophysics is a great example of biotech. Biophysics is really using physical tests to be able to answer biological questions. It's an example of biotech. So we don't offer that degree. And again, I don't recommend that you do a degree specialist in biotech. I recommend you do a broad degree. But how does that equip you to go and work in the biotech industry? Very well. We've got industrial placements between the second and the third year that students can elect to go on. They're competitive, but our students have succeeded to go to work for GlaxoSmithKline, for even accountancy firms, for many, many, many other firms. And you can select biotech firms where you can go and learn how to work in that environment. And you will see how the skill sets and the knowledge you are acquiring during your degree in Southampton helps you there. And that can equip you to work anywhere in the world. There is no bar. In fact, a degree from a Russell Group university will carry a lot of weight for you to then be able to work wherever you choose to work. So it's the work experience through the internships that will improve your prospects. And they can only look good if they come from a Russell Group university like Southampton, anywhere in the world, including India.
Great, thank you. Now, Netra asks a question. I think she's picking on from what you're sharing. What are your opinions on biomedical sciences as the undergraduate degree and then medicine for postgrad in the UK?
I think it's a brilliant idea. In the States, you can't do a medical degree without a first degree in biomedical sciences. And I really, really think that that is the better route because, again, medicine is an application of biomedical sciences. It's really learning about the body. Really, as a scientist, you understand how the cell gets sick and how that knowledge leads to treatment. And then as a doctor, you are applying that. Whereas if you go straight to do medicine, you're actually, it's like, that's why I left medicine after 2 months, because I didn't really want to just memorize which drug is used for which treatment. I wanted to understand the biology first and how that goes wrong. And if you did a degree in biomedical sciences, you could do a graduate 4-year medical degree. That's called a graduate degree. We offer that in Southampton. And if you get a first in biomedical sciences in Southampton, you will be guaranteed an interview in our medical school. The top 20 students who get a first are guaranteed an interview in our medical school to be able to do medicine afterwards. But you can do medicine anywhere. You don't have to do it in Southampton, but that's the link we have with our medical school.
Okay, then Nishiket Reddy is asking, which biological science degree would you suggest us to choose?
I think go with your heart, and one more arrow there will be microbiology. I think if you're not entirely sure what you want to do, I would go with a broad one like biology, because then you can specialize in anything. And that's another thing about Southampton. We have a lot of flexibility in our modules. So the first year of pharmacology, biomedical sciences, biochemistry, neurosciences, we also have neurosciences here. That's all the same. And the reason for that is you can come in to do biomedical sciences, which is also broad. And again, if you're not sure what you want to do, but you know it's got to be in the human body, choose biomedical sciences. Then at the end of the first year, you can specialize in anything. You can specialize in pharmacology, biochemistry, neurosciences, or you can just remain in biomedical sciences where you just learn a little bit about everything. So I would recommend that if you're not sure what you want to do a broad degree like biology or biomedical sciences. If you think that you want to be broad, but from the perspective of animals and not really that much about plants, then do zoology. If you are still not entirely sure, then take advantage of the fact that if you've got a broad degree like biomedical sciences or biology, you can tailor make the degree to suit your interests. So in the second and the third year, you only have 2 modules that are compulsory. Everything else is up to you. You study the module that takes your fancy. So no 2 people who do a degree in biomedical sciences or biology might actually leave with the same modules. They've taken the modules that satisfy their interests. So but my advice would be do the degree that excites you, do the topic that excites you, then you're much more likely to succeed. And choose a university where there's flexibility. We are so flexible that you can pretty much follow your heart in terms of not just the topics you want to study, but in our final year projects also equip you to be able to do the project either in biomedical research. Every university should offer you a final year project. It's a dissertation, a chance to do independent study. You can do it in biomedical sciences, or you can do a dissertation in science communication. You can do a dissertation in bioscience business, where you work with a pharmaceutical company, or you can do a dissertation in teaching where you can work with a school. So our flexibility is not just in the modules we offer, but also in the application of the modules afterwards and in the skill sets that you may need to then go into employability afterwards.
Okay, I'm going to club 2 questions. One is from Tanmay and the other is from Shaurya. They're both talking about the possibility of studying biochemistry or biology with business.
Mhm.
And so there are 2 different questions, but I think that's the overarching theme. It's if I want to join Southampton for BBA, which is business administration, can I have bio as a major or minor? And Shaurya says, if I plan on going into business after my undergraduation, should I do biochemistry degree or biomedical science degree? Would my choice affect my future career? So
very good questions. Both of them are very good questions, because what you're doing is you're asking, how does my degree help my employability prospects in the future, which is a very responsible thing to think about at this point. I would suggest that they both email me and I will find out for them whether they can do biology as a major and business as a minor, or business as a major and biology as a minor. If the major and minor degrees are where you do 2/3 in the major topic and 1/3 in the minor topic, and it just gives you the possibility of then learning about both. But if you wanted to actually think about doing a topic and then working in a business context in that topic. So for example, if you wanted to do biochemistry, and then you actually wanted to work in business afterwards to be able to work, I don't know, for a drug company where you want to really understand better the ability to market a drug, or pharmacology or biochemistry, you could do your first degree in pharmacology or biochemistry. Then I would advise you to do a master's in business, because it's during your master's that you learn the more specialist skills where you can apply the knowledge that you've acquired in your undergraduate degree to a more specialist area. So for example, I know somebody who did a first degree in biomedical sciences, and now she's an editor for Lancet, right? And how she managed that was she did her first degree in biomedical sciences, then she did a master in neurosciences, and then she specialized in science communication. And now she works with Lancet, where she's looking at how information that's not in the mainstream, health information that's not in the mainstream, like Ayurvedic medicine or tribal practices that are believed to have benefit for health, how they can be brought into the mainstream. So she does podcasts and she does specialist essays. She commissions specialist essays. So it's a very nice mix of many things, but the layering is done at different stages. If she had done a first degree in business, she would not have landed that job, because though she's understanding the business need to be able to tell businesses to take note of practices that may have health benefits, actually her main drive has to be an understanding of the biology for which there's a clinical need, for which the business has to now appear to facilitate the delivery to satisfy the clinical need. So I think be open, email me about majors and minors, and then think about, and I'm happy to, communicate with people after. Please take advantage of the emails. I will respond to you within 24 hours of receiving your email. And if I can't answer your question, I will put you in touch with someone who can, someone from the business school, for example. But that's what I would say. I would say, think about a broad degree, and whatever you want to work in, think about that as your master's, because that's what will then equip you with employers, even sometimes, depending on where you do your master's, where you can then apply that knowledge that can then lead to a job afterwards.
Awesome, thank you. There is a last question, which is more around your research. It's by an anonymous attendee, so I don't know if they're here or not. It says, beyond plaques and tangles, what other pathologies or mechanisms do you think are most critical in driving Alzheimer's?
Very good question. Inflammation. Inflammation is the culprit that sadly makes everything worse. So you could actually argue many diseases are inflammation gone wrong, because inflammation was supposed to be our friend that was supposed to turn up and clear those plaques and tangles. But when the plaques and tangles persist for so long, inflammation goes into overdrive. And that's when then it does damage in killing innocent cells when it's trying to kill cells that are full of plaques and tangles. So I think inflammation is the big driver, which is why I've just come back from a conference in Toronto, and they have found that the Mediterranean diet, which is full of anti-inflammatory things, and in India we've got turmeric, and you've got ashwagandha, and you've got moringa, all of these things are anti-inflammatory. But they have found that consumption of that kind of diet, which is full of anti-inflammatory foods, is very protective. So when your mums tell you to eat the rainbow and they give you haldi and in your milk, please do it. For us as well, we should be drinking that turmeric. So the driver is inflammation, and there's a lot of evidence for that. So anything that keeps inflammation at bay is going to be good for you.
Thank you, Professor. I'm going to just encourage you to leave your email in the chat box, or just bring that back on your screen so that students can just take a screenshot. Thank you for spending almost 70 minutes with us and talking about your research, the interest, various career opportunities, what can flow into another option and, become a very promising career. And I think we're all going back, leaving this webinar feeling enriched, and of course, remembering to have some haldi in all our food and, whatever we can. So thank you
No
very
problem.
much.
Yeah, and I will liaise again with Shreya and Nadeem about the other individuals who can talk about the road less traveled in the other areas we spoke about. So you can share that with whomever you want. But please do email me, and I will very quickly respond to your email and help you myself or transfer you to somebody else who can. Okay. Thank you very much.
Thank you. Thank you, all the students and counselors, parents who've joined us. We'll be back next Wednesday with another session. So this was the second in the series. We have 2 more to go, and I hope you'll be joining us to learn more about different careers as we go along. Thank you. Thank you very much.
Thank you.
Enjoy the evening. Bye. Thank you, Professor. Thank you, Nadeem. Thank you, Kasvi. Thank you, Sonali.
Thank you. Have a good evening.
Bye, Amrita.
Bye.
Bye.